Hello. How you doing? I should have the group exam grades, and I'll have all of your exams entering your grades hopefully today. So the average was a C, so the average is a C so far. Group exam grade will bring that up, so then I'll post final grades on Moodle. In case you're wondering, I don't have a disease or anything. It's Breast Cancer Awareness Month, so I told my daughter I'd dye my hair pink, and this is how it turned out. So the gray took up the pink quite well, the rest of it not so much. So let's talk about the heart. So we're going to continue in the cardiovascular system and the other systems that we talk about to also talk about the nervous system and action potentials, and to also talk about the endocrine system and those principles. So, yeah, hair's different. So we're going to go over those things. So if you kind of missed that stuff on previous exams, you get a second and a third chance. The neat thing about the heart is it is autorhythmic, means it generates its own rhythm. It doesn't have to be stimulated by the nervous system. The only thing the nervous system does is either increase or decrease the heart rate, or increase and decrease the force of contraction. You have cells that are electrically conductive cells. They're excitable tissue. They're similar to neurons, but not quite the same. They make up the electrical conduction system of the heart. So we have the SA node. We have these internodal pathways. So these are bundles of fibers, essentially axons, that go between the SA node and the AV node. They also spread out to form electrical synapses with cells that are contractile in nature. So the two types of cells we have are the electrical or the nodal cells, and those are more like neurons. And then the cardiac muscle cells that generate force, those are going to be more like skeletal muscle. The AV node, so the SA node, spreads depolarization through the atria by making electrical synapses with atrial, myocardial, or cardiac muscle cells. The information is transmitted via internodal pathways to the AV node, where it is delayed for about 1/10 of a second. And that's just to make sure that when the atria contract, the ventricles aren't contracting at the same time. So the delay in the AV node is so that when the ventricles contract, the atria are relaxing. You have then a bundle of His, which breaks up into bundle branch fibers. They have a left and right and some other smaller collateral branches, bundle branches, which terminate on Purkinje fibers, which will form electrical synapses with cardiac myocytes or skeletal muscle or cardiac muscle cells in the ventricles. So if we recorded action potentials throughout this entire system, this is what it would look like. Now, what we normally see is an electrical representation of all of those action potentials together called an electrocardiogram. That's how we can tell how the heart is working, if the electrical conduction system is working properly. So we have up here, the pacemaker of the heart is the SA node, generates an action potential. See how that's rounded? And that's because the channels that are going to be responsible here are calcium channels. So they're not the fast sodium channels that give a nice peak that we've seen with neurons. These are calcium channels. And then the atrial muscle Gives you this nice pointed peak and then a prolonged plateau phase. So the action potentials in cardiac muscle cells are going to be different than the action potentials in the electrical conduction system. Right? If you look at the AV node, you've got that same rounded calcium-dependent action potential. Then when we get to areas within the ventricular musculature, you see this really quick peak because of fast sodium channels with a nice spike where the sodium channels close, and then this long, drawn-out calcium plateau. So let's start at the beginning, which is going to be the SA node, the sinus node. That's the pacemaker of the heart, and it's the pacemaker because it's the group of cells that depolarize the fastest. So this is where the refractory period comes in. These cells depolarize and generate an action potential faster than the other cells in the conduction system do. Then they become the pacemaker because when the other cells are ready to generate an action potential, the SA node is already in the refractory period. So whichever group of cells depolarizes the fastest ends up being the pacemaker. So we have specialized cardiac muscle connected to atrial muscle. These are electrical synapses, so it's gap junctions we're dealing with and not chemical synapses, so there's no substances released. So an action potential generated in the SA node is immediately transmitted to all of the cells that it makes electrical synapses with because of gap junctions. The membrane potential here is going to be closer to threshold than the cells we've been talking about before. Right. The resting membrane potential for these cells is closer to what we talked about for smooth muscle. So when the membrane potential reaches threshold, which in this case is going to be about -40, calcium channels open, causing the action potential. So for the conducting cells in the heart, the rising phase of the action potential is due to calcium, not to sodium. And then after about 100 to 150 milliseconds, the channels close and you get potassium depolarization. Normally, your heart rate is around 70 beats per minute, which is slower than the spontaneous depolarization rate of the SA node. And that's because of the influence of the vagus nerve. Remember, the vagus nerve is a parasympathetic nerve. About 80% of the parasympathetic fibers in your body travel through the vagus nerve. So there's what's called vagal tone. So this vagal tone is keeping your heart rate at about 70, or lower than the spontaneous depolarization rate, which is actually closer to 100. Now the AV node cells have spontaneous depolarization as well, but they get stimulated via the bundle, the internodal bundle fibers before they reach threshold. So the SA node controls the rate of the heartbeat. If the SA node fails, then the ventricles will contract at the rate at which the AV node is depolarizing. So if your SA node fails, your heart rate drops to about 50 or 60 beats per minute, which is enough to keep you going, except if you decide to walk up a few flights of stairs. Then you're going to have trouble. Now, if the SA node and the AV node fail, the depolarization rate of the fibers, the Purkinje fibers and the bundle branch fibers, is so low as to not be able to keep you alive for very long. You're fine as long as you're not moving around and causing any oxygen demand for your tissues. And of course, as I said, the sinus node ends up being the pacemaker simply because it's the one that discharges the fastest. You end up with something sometimes called an ectopic focus, where a group of cells decide to go rogue and they're going off depolarizing and repolarizing on their own. That causes lots of problems, especially if they have a more rapid discharge rate than the sinus node. Then that group of cells becomes the pacemaker. So you end up with irregular beating of the heart and inefficient pumping of blood. Causes lots of problems. You can also get this when there's an AV block. So the SA node works fine. Something is blocking transmission through the AV node. And so you're going to get irregular beats. The beats of the atria and the beats of the ventricles are not going to match. So you're going to get inefficient pumping of blood. So if we look at the rhythmicity of these fibers, we start down here at resting potential. They have an unstable resting membrane potential. Right, said it's around minus 55, but it drifts. It's not stable. That's because there's a group, there's a type of channel found in these cells. They're found in some other cells in the body, but they're different than what we've looked at so far. They're called funny channels or hyperpolarizing channels, and they open when the cell gets back down to its resting membrane potential. So they open during the repolarization phase. And what they do is they allow sodium and some calcium to come into the cell slowly, such that the resting membrane potential is drifting towards threshold always. And of course, once it reaches threshold, then the calcium channels open, the voltage-gated calcium channels open. You get this spike, which is actually a rounded spike because it's calcium. As calcium comes in, when those channels close, then you'll get potassium repolarization. So you have this slow drift to threshold. You then have the calcium rising phase of the action potential, the falling phase or the repolarization phase because of potassium, and then that continues about 70 or 80 times per minute. So the number of depolarizing events that you will get from the SA node should equal the number of beats of your heart. Now, of course, you can't hear depolarization and repolarization. You can only record it. So that's why we do an ECG to see the electrical activity. But you can actually hear the heart as valves are opening and closing. So those are the sounds that you can hear through a stethoscope. Of course, ventricular muscle now is going to be different. So in this case, we've got a stable resting membrane potential, which is around minus 90. So it's far away from threshold, but it doesn't matter because these cells are connected by electrical synapses to the cells of the electrical conducting system. So any depolarizing event here is automatically transmitted to the muscle cells. So you reach threshold immediately. And that's what this very fast spike is, it's opening of voltage-gated sodium channels. The other telltale sign that this is a sodium channel is it's got this nice peak. Remember, sodium channels open fast, close fast. So you'll get this nice peak. Then there's a plateau because calcium channels, which were opening while voltage-gated sodium channels were opening, those calcium channels stay open longer, right? 100 to 150 milliseconds. So that gives you a longer refractory period for these cells. So when we compare the two, the electrically excitable or conduction cells and the ventricular muscle or the contractile cells, you can see the difference, right? You've got closer to threshold here, farther away from threshold here. You've got phase 0 for both, which is the rising phase of the action potential. And then you jump to phase 3 for the electrical conducting cells. There is no phase 1 or 2. Because phase 1 is closing of the sodium channels. Over here there are no sodium channels. And phase 2 is that prolonged refractory period. It's the absolute refractory period, but you also can call it the effective refractory period. Right? Remember, the refractory period is caused by sodium channels not resetting in neurons. And then once they're reset, the absolute refractory period ends. Then you have to deal with the relative refractory period, which is because you're hyperpolarized for a little while because of the slow closing of the potassium channels. In this case, the sodium channels have closed, but the calcium channels are still open. These are called L-type calcium channels. L for long. So they open slow, they stay open for a long period of time, and then they close slow. And once they close, potassium channels had actually started to open over here in phase 0, but because sodium and calcium are coming in, that overrides the effects of potassium leaving the cell. But once the calcium channels close, you get this phase 3 repolarization from potassium leaving the cell, which is the same over here in the electrical conductive cells. So the difference is, in the electrical conductive cells, you're closer to threshold. You have an unstable resting membrane potential. There's no phase 1 and 2, which are exclusive to the muscle cells, the contractile cells. The action potential, phase 0, is calcium. For the muscle cells, it's sodium. And then there's a calcium plateau that you won't see in the other cells. Then, of course, phase 4, we're back to resting membrane potential. Any questions? Okay, so in the ventricular muscle, you've got the sodium, calcium, potassium repolarization, and in the AV node, the SA node, the conduction system, the nodal fibers, you've got slow depolarization to threshold because of funny channels. Then got calcium for your action potential and potassium for repolarization. So if you were to tease all of these out of the heart, this is what it would look like without any atrial or ventricular muscle. This is the conduction system, SA node, internodal pathways to the AV node, and then a neat little anatomical feature called Bachmann's bundle. You might want to guess why that's there. Where's the SA node? What are the 4 chambers of the heart? right atrium, left atrium, right ventricle, left ventricle. So, yeah, Bachman's bundle. Make sure that the depolarization from the SA node travels to the left atria as well as the right. Because the SA node's in the right atria. It's near the entry point where the inferior and superior vena cava enter the right atrium. The Bachman's bundle transmits those action potentials to the left atrium, so that both atria are contracting at the same time. You have an AV nodal delay, a bundle of His, which I call the Slytherin bundle. Breaks up into left and right bundle branches. So you got a left branch for the left ventricle, right branch for the right ventricle, and then Purkinje fibers project off of that and form electrical connections with the cardiac myocytes, the muscle cells. Now there's also this left posterior bundle, which branches off from the left bundle branch to make sure that it goes around and contracts the backside of the heart. So the ventricular musculature of the left ventricle is much thicker than the right ventricle. So there are more cells to excite. So you have to have this extra set of branches to make sure the entire left ventricle is contracting somewhat in unison, or at least the way it's supposed to. And I don't know why I threw that in there. Yeah, so there's 3 internodal pathways: anterior, middle, and posterior. So the AV bundles, they can contract in both directions, but just like an axon can. But since you've already got an action potential traveling in one direction, it sets up a refractory period behind it. So you don't normally get transmission backwards. It's almost always going in the same direction in normal conditions. Pathological conditions can cause, so if you have an ectopic focus or some type of heart block, you can then get retrograde transport of action potentials, but it's not normal. So these bundles are the only conducting path between the atria and the ventricles. We talked about left and right bundles. And the transmission time is about 0.06 seconds, which if you look on an ECG, it correlates somewhat to the QRS complex. The Purkinje system, sometimes this is referred to as the His-Purkinje system. I know they like to do that in anatomy. I'm kind of separating it out. Anybody lose sunglasses? No? All right. So the Purkinje fibers are the ones that are going to be making electrical synaptic contact with the muscle cells that are going to contract and generate force and pump the blood. So they have a very fast conduction velocity, lots of gap junctions. That's where you're going to find those intercalated discs that we talk about with cardiac muscle. So the transmission of the electrical impulse through the heart starts at the SA node. Transmission then travels through the internodal pathways to the AV node, at the same time also traveling to the atrial muscle so that it contracts. AV nodal delay. The AV node is going to be slow to depolarize, so there's going to be a delay of about 1/10 of a second. So the atria have time to contract and start to relax before the ventricles get depolarized. So all of these events occurred during the P wave of the electrocardiogram. Then when the AV node starts to signal the His bundle, the two bundle branches, the Purkinje fibers, then you're going to start to see the QRS. So you're going to get depolarization then of the ventricles. And you're going to try and make it so that the heart contracts from the apex, the pointy end, towards the base, which is where the atria are. Because the aorta is up here, coming out of the top of the heart. So you want to push blood in that direction. So if you start contracting from the base down, it's going to be inefficient. So that's why these bundle branches travel through the interventricular septum here, and then they start the actual act of making gap junctions and contracting muscle cells down towards the apex. And then they're all connected by gap junctions, so those intercalated discs are going to allow for depolarization to spread throughout the entire ventricular musculature relatively rapidly, so it all contracts as a unit. I'll review from anatomy, right? You all remember this? Kind of. So the current spread, every time I say spread, I think of football. The current spread here, if you relate this to the electrocardiogram tracing, you've got your P wave when the atria, when those atrial cells are depolarizing and repolarizing. So the initial depolarization gives you the upswing in the P wave, and then after total depolarization, when they start to repolarize, you get the downward portion of the P wave. The atria are relaxing. The AV node sends the signal through the bundle of His, the bundle branches, to the Purkinje fibers. So you'll start to get this Q wave. And then as the wave of depolarization travels throughout the entire ventricular myocardium, you'll get the R and the S, so the whole QRS complex. And then the depolarization of the ventricles happens during the T wave. So you can see this interval between the P and the QRS, this area here, is going to be when the atria are contracting, and then from this QRS to the T here is going to be when the ventricles are going to be contracting. So we can divide this ECG up into segments or intervals. Right, the PR segment from the end of the P to the Q, PR interval from the beginning of the P to the Q. Right, so this PR interval represents atrial systole. Remember, systole is when the heart is contracting. Diastole is when it's relaxing. And then this QT interval here, from Q over to T, is going to represent ventricular systole. You with me so far? Any questions? Yes?
What was the difference between the PR interval and the PR segment?
The PR interval is the entire atria. This PR segment actually only represents the muscle cells. And that's the kind of similar over here. The ST segment doesn't tell you much. It's more the QT interval. So some of the things we can take away from EKGs. P waves should always immediately precede atrial contraction. The rule in physiology is that electrical events happen before mechanical events. The QRS then should appear immediately before ventricular contraction. The ventricles will remain contracted after the T wave. So while they're repolarizing, they start to relax. Remember, muscle cells contract because of what? What enters muscle cells to cause them to contract? Calcium. So to get them to relax, you have to remove calcium from the cytoplasm. So even though the cells have repolarized, there's still cross bridges that are formed. So you have to get rid of the calcium to break up those cross bridges. So the atria remain contracted until they are repolarized. You won't see, however, the U wave, the repolarization, because it occurs during the QRS, so it kind of gets drowned out. You can't record two waves at once. The P to the Q, the PR interval, lasts about 0.16 seconds. And that's going to be the spread of depolarization throughout the atria, and then atrial contraction. And then the QRS is going to be the beginning. It's going to be the depolarization, and then the QT is going to be ventricular contraction occurring during that phase. And that's going to last longer. That's about 0.35 seconds. So if you add them all up, so if you say that you've got 0.16 seconds, and then you've got a 0.1 second delay, then you've got 0.35 seconds, and then there's a space of time between the T and the next P, you end up somewhere around 0.8. So every 0.8 seconds, you're getting a signal for a heartbeat. The recording that we have, the electrocardiogram, that occurs because you're 70% water, and water conducts, especially if it's got ions in it like sodium and magnesium and calcium and chloride. So the electrical signal generated by the heart can actually be read through the interstitial fluid throughout the body. So all those action potentials that are occurring get summed together, and you can measure them with a group of sensitive electrodes simply by placing them on the skin in certain parts of the body. When you get a wave of depolarization passing between two electrodes, you get an upswing on the trace. So that's going to represent depolarization. And then after that wave has passed, you've got no electrical signal between these two electrodes, so you get a return to what's called the isoelectric line, or the isoelectric point, where there's no signal. And then when repolarization occurs, you'll get a downswing, because now you've got a negative signal traveling between those two electrodes rather than a positive signal. And so this kind of represents, if you could take the atria and record it all on its own without the ventricles, this is what you would get. You would get a P wave, then you would get a repolarization wave. But on the ECG, at this repolarization wave, you get the QRS. So you'll never see it unless there's some pathological condition. You can use this information to calculate heart rate. Heart rate can be measured in a variety of different ways. You can look at pulse rate. You can do auscultation, which is putting a stethoscope on somebody's chest. Or if you take an electrocardiogram, you can measure it as well. So what you do is you take the reciprocal between the two QRS complexes. So you measure from the peak of one R to the peak of the other R, normally about 0.8 seconds. So 0.83 seconds here, and the reciprocal of that, so 60 over 0.83, gives you a heart rate of 72 beats per minute. So that's a very simple calculation that you can do. So if I give you an electrocardiogram, you can figure out a lot of information from that. You can tell what's going on in the atria, you can tell what's going on in the ventricles, you can come up with a heart rate, you can tell if the heart is skipping beats, you can tell if there are extra beats. A lot of information you can get from that, which you will talk about in the cardiology unit, because that's all disease states. I don't go through that here too much. What I will talk about is heart block. Different types of heart block. Heart block is basically something is interfering with transmission through this electrical conduction system, either from the SA to the AV, from the AV to the ventricles, bundle branch block, all these other things that could occur to prevent electrical signals from getting to the cardiac muscle cells. In first degree heart block, the impulses are slowed, but they eventually reach the ventricles. This is not life-threatening. People can live long lives with this. And actually, some athletes may have it, and you may see it show up as side effect for some medications. It's not usually a problem. Second degree heart block, what is called type 1. The impulses get delayed further and further with each heartbeat until you eventually get a period of time where it skips a beat and then resets and then goes back, and then there's a delay for each heartbeat and then a skipped beat. That may be asymptomatic. You may have that for a long period of time and not know about it. May cause some dizziness, some difficulty during exercise. Second degree heart block type 2, that's when we start to get a little more serious. Some electrical impulses are absolutely unable to reach the ventricles for whatever reason, whatever the pathology is. So you end up with skipped beats. And the problem is type 2 can easily progress to type 3. Type 3 is called complete heart block. So none of the electrical impulses from the atria are reaching the ventricles. So you've got the atria contracting and relaxing at their own rate, say 80 or 90 times per minute. Then you've got the ventricles contracting only because of signals from the AV node at 40 or 50 times a minute. So you've got ineffective, it becomes an inefficient pump. You've got ineffective pumping. In this case, patients are going to feel really sluggish, really lightheaded. They're going to have a lot of difficulty with any type of demand on the cardiovascular system. So even going up some steps places too much of a demand on the cardiovascular system for them. And a bundle branch block, when you get complete block, impulses are slowed or blocked to one or both of the ventricles. Now, all of these will show up on an electrocardiogram. Right, so if we've got our normal here, first degree heart block, you can see the PR interval is longer. Right, the impulses have been slowed. So you've got slowed transmission. So you're going to get an abnormally long PR interval. When it progresses to second degree, now you're going to start to skip beats. See here between these two P waves, there's no QRS. And the QRS looks funny. Doesn't look like it does up here. All right, so transmission through the conduction system has been further hampered. There's something called a Weichenbach phenomenon, where you've got differing lengths for PR intervals, and you've got missing heartbeats. Complete heart block, you're going to see lots and lots of P waves, and every once in a while you'll get a QRS. Right? So complete heart block, the SA node is controlling contraction of the atria. The AV node here is firing on its own. It's going rogue. It's got no parental supervision from the SA node. So you're going to get many more P waves than you'll get QRS complexes. So there's a little heart block poem to help medical students and nursing students remember this. So if the R is far from the P, then you have a first degree, right? Extended PR interval, first degree heart block. Longer, longer, longer drop, then you have a Winkenbach. Just, you can memorize this and say it down at Tom's. It's kind of fun. People think you're nuts. If some P's don't get through, then you have a Mobitz 2, which is just a regular type 2 heart block. Right? You can see the one skipped beat here. Up here in the Winkenbach, you see the extended PRs until you get this one big jump between QRS complexes. Then if the P's and Q's don't agree, then you have a third degree. There's more P's than Q's. Questions? Yes, sorry.
So is Winkenbach like type 1 and then the Mobitz is type 2?
No, they're both type 2. They're just different types of type 2.
So is it like type 1 and type 2? Because we had like second degree and you have type 1 and type 2 second degree.
Mhm.
They're both second degree. One's type 1 and second degree. One's type 2.
Yeah, there are different types of type 2. I wouldn't call it type 1, type 2, type. That gets too confusing. So just say type 2 and Winkenbach. They're both type 2s, it's just that Winkenbach is specific. All right. So these are electrically excitable cells, so they are susceptible because their resting membrane potential is determined by potassium across the membrane to changes in potassium. Now, hyperkalemia is much more severe than hypokalemia. What mechanism do we have to correct hyperkalemia? Aldosterone. Right? The primary stimulus for release of aldosterone is high potassium, hyperkalemia. So aldosterone causes you to get rid of excess potassium in the urine, reabsorb sodium in the process to maintain electrical balance. So we've got a mechanism to protect for hyperkalemia. What protects you from hypokalemia? We've got nothing. So that right there tells you whoever designed the system thought hyperkalemia is more severe than hypokalemia. So in hyperkalemia, you're going to start to see these really big T waves. And then once you get up around 8 to 8.5 for a blood concentration of potassium, you're going to start to see the Q wave either split off and become bimodal or disappear. Hyperkalemia, severe enough above 8.5 can cause heart failure. The electrical conduction system just can't generate impulses anymore because of the severe hyperkalemia. Hypokalemia, on the other hand, the T wave will start to split off from the U wave. So you'll see an extra wave there. And then if it gets severe, the U wave will kind of drown out the T. Hypokalemia can become severe below 2.5, but you're usually going to see problems with other systems, the brain, skeletal muscle, before you see problems with the heart. Parasympathetic effects on heart rate. Parasympathetic system slows heart rate because of its effect on muscarinic type 2 receptors, right, that we've gone through. Hyperpolarization, moving those nodal cells farther away from threshold, are the effects of acetylcholine. So transmission of impulses may temporarily stop the heart rate. if you've got a mass discharge down the vagus nerve, you can get a brief stoppage and then re-engagement. But when that occurs, you end up with what's called ventricular escape. Right, so excessive vagal discharge that causes your heart to stop beating, to skip a beat. So if your heart rate falls below 40, because of the AV node, you're going to get ventricular escape from this phenomenon. But of course, because of the low heart rate, you're going to get low blood pressure and dizziness, fainting, which is called syncope. So these effects here, prolongation of the slow depolarization period is one way in which you can slow heart rate. Another way is to hyperpolarize, so get a negative shift so that phase 4 moves farther and farther away from threshold, so it takes longer to depolarize. That's going to slow heart rate. Or you can change the threshold, which is not the most common. So if you move threshold further away from resting membrane potential, that's another way to decrease heart rate. Sympathetic increases heart rate because of its action on beta-1 receptors. Right, so beta-1 receptors increase cyclic AMP, muscarinic receptors decrease cyclic AMP. So they use the same second messenger system, just with different effects. Right, so sympathetic nerves release norepinephrine. You're going to get an increase in heart rate. You're going to get an increase in rate of conduction of the impulse through this electrical conduction system. And you're also going to get an increase in force of contraction because of an effect on the ventricular myocardium. So with the parasympathetic system, you're simply affecting heart rate. You're not really affecting force of contraction. With the sympathetic system, you're affecting multiple things. So you've got heart rate, you've got force of contraction, rate of relaxation, a number of other things. Inotropy, we talked about inotropy. So inotropic agents are going to change the contraction force of the heart. Chronotropic agents are going to change the heart rate. Dromotropic agents are going to change the conduction velocity or speed through the bundle. Lusitropy we're not going to talk about. That's the rate of relaxation. That's a little bit more advanced. That confuses things. So if we look up here, you've got inotropic, right? Alpha 1, beta 1, and beta 2 all respond to norepinephrine. Chronotropic, same thing. Dromotropic, same thing. Right, and then acetylcholine, really not affecting as many things as we get here. Keep in mind, the alpha 1 on vessels is for constriction. There is no alpha 1 on the heart. The beta 1 is for constriction on some vessels, very few. Beta 1 is the receptor you find on the heart. Beta 2 is found on vessels or in the lungs. So overall, we have to go back and revisit the autonomic nervous system when we talk about cardiovascular function. Because the alpha receptors control peripheral resistance. They control the diameter of most of the blood vessels. Alpha 2 receptors are more for controlling the autonomic nervous system, particularly the sympathetic branch itself. They're autoreceptors, so they inhibit norepinephrine release. So if you get excess norepinephrine in the synapse, they stimulate alpha 2 receptors to decrease release of more norepinephrine. Beta 1 receptors give you tachycardia. Right, stimulation of beta 1 receptors are going to increase heart rate. Beta 2 receptors are mostly for dilation, smooth muscle relaxation, so they're found in the lungs, in the coronary arteries, etc. Any questions? See you on Wednesday. There's a homework for Wednesday, the 11th. You'll be gone. Monday on break. Have fun.